I'm going to skate across antifungal resistance mechanisms and drivers, and I'm essentially setting the scene for Joe's talk next. He'll be drilling into some of the genomics we use to track and trace resistance. But what are fungi? They're just it's a wonderful kingdom, the fungal kingdom. Billion years old, used to be swimmers back in the day, invaded land 500 million years ago, and that catalyzed the invasion of land of plants as well. Once there, they radiated into the higher fungi, the dikarya, and it's just there's upwards of 9 phyla within the kingdom. We've described 5% of the 2.5 million species that we share the planet, and buried within this gorgeous kingdom, there's some absolutely, horrendous pathogens. So when we're thinking about vertebrate disease, the Ascomycota, the molds, contain 90% of human infections, but then those mushrooms that you like to buy and eat from the supermarket also contain some heavy hitters there, specifically the yeasts, the cryptococcal yeasts. So Nicola is going to talk about the plant pathogens, so I won't burn her space on that one. But this omnipresent miasma of fungi that we share the planet with, means that we've always had to defend ourselves against their ability to degrade our food security and also just to predate upon us. So, originally we used salts of various sorts to knock down, to steal water and knock down the mold action. And then through the 18th and 19th century, we became more innovative in using copper solutions to act as antifungals. And then in the 20th century, we had this explosion of antifungal chemicals that were discovered both in the plant and animal space. There's at least 25 different mode of actions in plant antifungals, fungicides. With animals, we're limited to 4 categories of animal antifungal class modes of action, starting with the polyenes, which were first discovered in the 1950s. And the world doesn't stay the same. There's been innovation both in agribusiness, but also in the clinical medicine, and we now have new mode of action antifungal chemicals coming. So there's a lot of innovation in this space. Now, the reason that we need all this innovation is fungi are complex eukaryotes, and they are, they're microbes, so they respond in a microbial manner to selection, strong directional selection in the environment, but they're also eukaryotes, so they do it in a very different way to the bacteria that some of you will be more classically focused on thinking about. And there's a number of features of the kingdom which allow them to respond, really rapidly to strong directional selection produced by our efforts to contain and control them. You've got variable mutation rates that can go very high. As those who are familiar with your moldy homes, they produce this vast inocula. Of course, that translates into very high effective population sizes. So there's a lot of heritable variation. If you look at that bottom left-hand, this diagram, we have a limited set of targets that we can use to attack the fungal genome, because of course they're eukaryotes. So that means that the chemicals we use are limited, but they're probably going to be static rather than cidal. So we're into the region of having to give long-term therapy, repeated treatments, and we grow monocultures. So there's a huge amount of space here for the evolution of resistance to occur. So yes, we have all these antifungal drugs, but as Darius has said, resistance has evolved to all of the classes that we've used. And there's a broad variety of mechanisms out there, which I'll mention in a subsequent slide. But because of this limited opportunity for antifungal drug development, you often have innovation that's it iterates on the same class of chemicals, both in the agricultural and human space. So that's the phenomenon of dual use. So let's just kind of think about comparing drivers of bacterial and fungal AMR, because it's important in understanding how we can cross-inform tools across between the prokaryotes and the eukaryotes. They've got low fidelity species boundaries in bacteria, so widespread horizontal gene transfer. And this doesn't occur to that anything like that extent with fungi, who are, very choosy about who they have sex with. Lots of reproductive barriers there. So these porous gene boundaries between bacterial species means there's a lot of transfer of mobile genetic elements and hitchhiking. So if you're worried about resistance, you've got to track those. But that's not important to anything like that extent, because you don't have HDGT and hitchhiking on that scale. For bacteria, resistance does not devolve de novo for each species drug combination, because of course you can have this broad porous transferability, whereas it does have to evolve de novo for each species drug combination that imposes limits on evolutionary rates. Bacteria have extrachromosomal plasmids constituting an enormous pangenome that contain the majority of ARGs. These extrachromal elements do not exist to that extent in fungi. Zoonotic human infections by AMR-rich pathogens are a feature for bacteria, currently unknown for fungi, and antibiotic resistance has been a long-term target of international study pretty much since Fleming's time. You have wonderful systematic surveillance, reference laboratories, exquisite genomic tools. Believe me, I've watched them over the years. Antifungal resistance is a very new phenomenon. Bacteria have enormous funding, and here, playing the tiny violin, the fungal field has microscopic funding in comparison. So those are the differences. What are the similarities? Bacterial AMR got active global spread through travel and trade. Yes, we have this with fungi. Nosocomial healthcare outbreaks are a feature of bacteria, gut, mucosal and skin commensal carriage. Yes, this is also a big problem with fungi in healthcare settings. Subtherapeutic levels or inadequate exposure to antibiotics can drive resistance. Yes, fungi. We have enormous use of antibiotics in agriculture. We have enormous use of fungicides in agriculture. Same. And there are probably many similarities in resistance mechanisms, copy number variations, SNPs, so on and so forth. We see parallels for that in fungi. So this is the sort of space where we can compare and contrast across our systems. So how does a fungus acquire resistance? So I'm not going to get into the weeds with this particular comparison. Go to a fungal genetics conference and it'll blow you apart, because they're so sophisticated as organisms in evolving resistance. But certainly pumping the drugs in and out, overexpressing the drug target, evolving SNPs, mutation rates are important, and then just booting up chromosomes through heteroresistance or aneuploidy, copy number variation are very, very important in building resistance to azoles. There's target the cell membrane. Kinocandins, the class of drugs that target the cell wall. Again, you have SNPs are very noticeable and easily recognized in the key genes there. But then you're into this wealth of epigenetic responses that we're only beginning to understand that confer resistance. So, obviously you can track and trace SNPs and polymorphisms and tandem and promoter mutations with genomics, but this epigenetic response where heat shock response confers transient resistance, much harder to get into. Polyenes also the mainstay features of defeating serious fungal infections. Here we again have a lot of epigenetic factors at play, and very little polymorphisms and mutations that we can track and trace with resistance. But resistance is very hard to evolve with amphotericin and so on and so forth, so less of a problem there. And flucytosine, these are the pyrimidine analogs. They actually inhibit DNA RNA synthesis, and again mutations, polymorphisms that we can track and trace. So broadly, that's where we are with understanding resistance to our current armamentarium of 4 classes of antifungals. But then we've got these wonderful new mode of action classes, the charge really being led by olorifim and fosmanogepix, both in late phase 3 trials. And you talk to clinicians who have given these drugs to patients who have got resistant infections from either Coccidioides immitis or Aspergillus fumigatus, and those patients which are absolutely finished recover and are with us today, completely cured. So these are miracle drugs. However, resistance will evolve. So yes, fungi are the Cinderella compared to bacteria, languishing in the shadows. This is recently changed by the WHO fungal priority pathogens list, where they've name-checked these particular fungal pathogens as high priority targets. Billions are infected, millions per year of fungal infections, equating to a burden of mortality up there with malaria and TB. No vaccines, few drugs. And of all of these organisms, Aspergillus fumigatus explains probably half of the global problem in our eyes in terms of burden of mortality and arguably morbidity. However, we have resistance emerging across a number of these, and you can see it stacked up in that critical and high group. This has been recently put together by Joe Rhodes. It's in Press in Nature Microbiology, and you can see that this isn't a background level of resistance, it's an Anthropocene increase. So why is this? Let's just think about the azoles. We're addicted to this class of chemicals. It's used enormously in agriculture. It underpins food security across the planet. And you can see this year-on-year increase through the in the United States. Since the introduction of this class of chemicals in the 1980s, we've seen resistance accruing in a vast number of different pathogens, both in the plant and human space. This is the usage per ton across Europe. It's relatively steady, but the main point here being that we'll use 1,000 times more azoles in agriculture than we do in human health. So this has led to the now very well recognized problem that resistance to Aspergillus, the most important fungal pathogen to which we're all exposed, evolves in nature. So you can evolve resistance when you're on long-term prophylactic therapy, just through exposure in the lung. But the first cases of resistance were seen in the 1980s, and then the observation that resistance was occurring in nature. So since these original papers, there's been the whole kind of drive to better understand this resistance in the environment, and it's first noticeably different to the sort of resistance that you'd evolve in a patient's lung. When you see azole-resistant Aspergillus in the environment, there's a tandem repeat in the promoter region and some SNPs, and these accounts for well over 90% of the resistance that you'd see occurring widely in the environment. So over the years, we've been sequencing our way through Aspergillus across planet Earth, both from the clinic and the environment. And it's become very obvious that it's everywhere, and that also resistance is very stacked into a cluster within this global phylogeny. So we call this cluster Clade. And you can see there, you don't need to do fancy statistics on that. The resistance is highly clustered. And there's the tandem repeats 34 and 46, which mark environmental resistance. Now, the remarkable thing about this Clade is not only is it very highly resistant to the azoles, but it's also resistant to drugs that we've used in the environment that are now banned. So it's immortalized the ghost of previous fungicides, the benzimidazoles, that's all stacked up in Clade, and the cytochrome quinone out. These are used contemporary at the moment, but you also have the resistance here. So this is an extraordinary cluster because it has evolved resistance to 3 different, entirely different mode of action antifungals. So it really is a monster. The light bulb moment, in my eyes at least, was this work by Mike Bottery up in Manchester, where he has shown very convincingly, this is now published in Nature Communications, that clade A is a mutator. It's not a hypermutator. The mutation rate is dialed up about fivefold, but it's enough to make sure that when you're in a selective environment, that this particular cluster evolves resistance before other wild types. So this is a really interesting model to play around with. It's not just Aspergillus that we see elevated mutation rates allowing resistance to develop. We showed that this had occurred in cryptococcal meningitis, where mutator phenotypes of this particular yeast evolved resistance within these long-term human fungal infections. So it seems that there's many different there's a distribution of variable mutation in the environment, but it's those mutators that evolve to patient. And there's a very big phylogenetic component to this, both in Aspergillus and Cryptococcus, and probably most fungal pathogens. So this is the conjecture. Aspergillus fumigatus mutator alleles occurs at a background rate in the environment. Got a very strong selective environment. So these mutator A genotypes respond to directional selection and sweep up in frequency as they pick up polygenic resistance to these complex agrochemical cocktails. And eventually that you end up with something like clade A. So here you go, the Pandora's box of mutation, changing agrochemical use, and the hitchhiking of the different resistance on that particular haplotype, and that's where we're going with fumigators. So will it stop? Will it end up with clade A fixing in the environment? That's the question. So it's not just Aspergillus fumigatus. We have burgeoning multi-drug resistance fungi. There's the pandemic of Candida auris. It's a creature from the Black Lagoon, says Tom Chiller, chief of the mycotic diseases and CDC. And this is becoming a real issue across the planet. Here you see the increase in healthcare settings across the United States. And here we're actually seeing spill back into the environment as Candida auris is now establishing widely in feral dog populations. So, you know, it really is going to end up becoming a dominating yeast across the planet. This is a really interesting one, pandemic Trichophycon indotinia. This one isn't going to kill you, but it is going to make you very, very unhappy because it causes this broad inflammatory reaction across the whole body as it spreads, and it isn't resistant to terbinafine. So its emergence, and you can see the increase here, is linked to the topical use of steroids and antibiotic creams in very high density urban populations of the world, and principally this appears to be Pakistan, India, and Bangladesh. And if you have this particular dermatophyte, you have to take oral anti-hydroconazole to clear it. This is becoming a real issue in the UK as we're seeing this increase in Trichophycon indotinia in our populations here. So obviously there's a real need to be able to use genomics to differentiate between these different dermatophytes, to look for pathways of transmission and actually do the epidemiology that's necessary to understand the emergence of this particular resistant and highly transmissible organism. So a lot of work going on behind the scenes with UKHSA on this one. I mean, more broadly, you can see where I've been going with this. There's a global need for One Health roadmaps to better understand these patterns and processes. Fungal antimicrobial resistance is a very environmental problem, both linking together the ways that we farm, but also what we'd actually do to ourselves. and how we you know, in terms of trying to manage gigantic populations of humans. And I haven't mentioned those new patient populations, people with COVID-19 who are increasingly predated upon by fungi in the environment. So we need better diagnostics to detect. We need better genomics to be able to associate patterns and processes. And this is all the sort of generating the baseline data that we're going to need to impact policy, because that's what's needed in order to do anything against the rise of genotypes such as clade A Aspergillus fumigatus. So yes, I've got 10. Oh yes, basically we need to move the global needle on fungal antimicrobial resistance. And in that wonderful UN General Assembly focus on AMR, there was no mention of fungi. So we can do better.